So today in this topic we are going to start the microbiology uh, chapter. So in the microbiology we are going to start from the basic bacteriology. So in the basic bacteriology today in this video we are going to talk about different bacterial structures. So we are not going to talk about the different bacterial organelles because the bacterial structures are the more important ones for their action, for their microbial activity. So we'll talk about various appendages, the specialized structure, and the cell envelope. Here you can see the figure of the cell envelope of the bacteria. Here you can see the cytoplasmic membrane. The cytoplasmic membrane is same as the cell membrane of our body, which is the bilipid layer. So above the so it is the here is the figure of the gram-positive bacteria, the cell envelope of the gram-positive bacteria. Here it is the gram-negative bacteria, gram-positive and gram-negative. So if you see both in the gram-positive and gram-negative bacteria, there is the cytoplasmic membrane. The cytoplasmic membrane in the both gram-positive and gram-negative bacteria are the bilipid layer. So above the cytoplasmic membrane, so you can see. So if you think it is as a bacteria, so if you think it has a nucleus, so we are taking the cross section of this part. So we are taking the cross section of this part. I mean, we are taking the detailed structure of this part. So detailed structure of this cell envelope. So this detailed structure of this cell envelope is projected in this figure. So you can see the cytoplasmic membrane is the bilipid layer in both gram-positive and gram-negative bacteria. So above the cytoplasmic membrane, there is the cell wall. The cell wall is made up of the peptidoglycan. And above the cell wall, in the gram-positive bacteria, there is the capsule. You can see here the capsule. But in the gram-negative bacteria, it is a little bit unique. Above the cell wall, you can see the another layer, that is the outer membrane is present in the gram-negative bacteria, which is unique to the gram-negative bacteria. So so after the outer membrane, there is only capsule is present in the gram-negative bacteria. So if we look at the cell envelope in the gram-positive bacteria, there is cytoplasmic membrane, and above that cytoplasmic membrane there is the cell wall, and above that cell wall there is the capsule. And in the gram-negative bacteria, you can see there is the cytoplasmic membrane, and above the cytoplasmic membrane there is a peptidoglycan layer, and above the peptidoglycan layer there is one outer membrane, and above that outer membrane at the last there is the capsule. So it is the cell envelope of both gram-positive and gram-negative bacteria. So now let us discuss detail about the various appendages, specialized structure, and cell envelope of the both gram-positive and the gram-negative bacteria. First of all, let us discuss about the flagellum. So if you look at the flagellum, this flagellum is originated from the cytoplasmic membrane. So you can look in this flagella. This flagella is originate from the cytoplasmic membrane. So this flagellum, so this is the flagellum. So both flagellum of the gram-positive and gram-negative bacteria. So this is the chemical composition of the flagellum is it is made up of protein, and the main function of the flagellum is the motility of the bacteria. Now let us discuss about the pilus. So the pilus, you can see it is both present in gram-positive and gram-negative bacteria. So the chemical composition of the pilus, or it is also known as the fimbria, is the glycoprotein. So the main function of the pilus in gram-positive and gram-negative bacteria is it mediates the adherence of the bacteria to the cell surface. So and also the sex pilus forms during the conjugation. So the detail about the conjugation we will talk that in detail in the bacterial genetics. Now let us move towards the specialized structure. I want to give emphasis on the spore. What is spore? So as you know, the chemical composition of the spore is that the spore is the keratin-like coat in the bacteria, it is dipicolinic acid, and it is the peptidoglycan, and mainly is composed of the DNA. So the function of the spore is, it is mainly associated with the gram-positive bacteria only. So the mainly the spore is essential for the survival. So the spore mainly helps in resistance of the dehydration of the bacteria, and it also resists heat and the chemicals. So basically the spores protect the gram-positive bacteria against the dehydration, heat and chemicals. So spore is very much important for the gram-positive bacteria. Now let us talk about the capsule. The capsule of the bacteria. The capsule is present in both gram-positive and gram-negative bacteria. You can see the outermost layer of the both gram-positive and gram-negative bacteria is the capsule. So capsule is the discrete layer usually made up of polysaccharide, and rarely it is made up of protein. So the main action of the capsule in the bacteria is to protect against the phagocytosis. So the capsule is the structure that protects the bacteria against the phagocytosis. Now let us discuss about the slime layer. Slime layer. So the slime layer is the loose network of the polysaccharides. The slime layer mediates adherence to the surface, especially the foreign surfaces. So for example, if we keep the indwelling catheters or any other foreign surfaces, then the slime layer will help to mediate the adherence to those foreign surfaces. Now let us talk about the outer membrane. The outer membrane of the bacteria. As you can see, the outer membrane of the bacteria is unique to gram-negative bacteria only. So this whole portion is the outer membrane. As you can see, the outer membrane is not present in the gram-positive bacteria. It is only present in the gram-negative bacteria. So below the capsule layer, the outer membrane is present in the gram-negative bacteria. So outer membrane has the two parts, the outer leaflet and the inner leaflet. The outer leaflet, you can see, this is the, sorry, I'll use another marker. So it has the this whole portion is the outer leaflet, and this is the inner leaflet of the gram-negative bacteria. So the outer leaflet, you can see, it contains the endotoxin. So these are the various endotoxin, or we call it is the lipopolysaccharides. It is embedded protein and porins and other outer membrane proteins. So the inner leaflet, you can see the inner leaflets are the phospholipid in the gram-negative bacteria. So it is unique to the gram-negative bacteria only, and the endotoxin that is present in the outer membrane. So it is the lipid A induces the tumor necrosis factor and the interleukin-1. The antigenic O polysaccharide components are the most antigenic. So the porins that are present in the outer membrane are transported across the outer membrane. The porin that you see, so this is the porin in the outer membrane, will help to transport the various substances outside the outer membrane in the gram-negative bacteria. Now let us talk about the periplasmic space. So this is the periplasmic space. So the periplasmic space, you can see, is the space between the cytoplasmic membrane and the outer membrane in the gram-negative bacteria. So there is peptidoglycan in the middle, you can see. And the main function is that it is the net-like structure which gives the rigid support, and it protects against the osmotic pressure damage. So this mainly periplasmic space helps the bacteria to be protected against the osmotic pressure damage. Now let us talk about the cytoplasmic membrane. The cytoplasmic membrane is the phospholipid bilayer sac with embedded proteins like penicillin binding proteins and various other enzymes. The lipoteichoic acid, you can see the lipoteichoic acid is unique to gram-positive bacteria only. So the lipoteichoic acid only extend from the membrane to the exterior. So you can see here, and the main function of the cytoplasmic membrane is that it is the site of oxidative and transport enzymes. So you can see the lipoteichoic acid induces the tumor necrosis factor alpha and interleukin-1. So these are the main functions of the lipoteichoic acid. So we discussed about the various bacterial structure. Again, I am going to tell you, in the gram-positive and gram-negative bacteria, both have the cell membranes, and above the cell membranes both have peptidoglycan layer, and above that peptidoglycan layer, the gram-positive bacteria has the capsule, but the gram-negative bacteria has the outer membrane, and above that outer membrane only there is the capsule. The key two differences between the gram-positive and gram-negative bacteria are: the gram-positive bacteria have the lipoic acid, and the gram-negative bacteria has the outer membrane, which is unique to itself. So I hope you understood this topic on the various bacterial structure. We talked about the various appendages like flagella and pilus, and specialized structure like spore. We talk about the cell envelope, which consists of the capsule, slime layer, outer membrane, periplasm, cell wall, and cytoplasmic membrane. So I hope you understood this video on the various bacterial structures. Thank you.